Okay, good morning everyone. So I hope you are doing well at home. You are safe there at class. So for today's lesson, we will tackle about the lesson 2 in module 2, which is the kinetics of fermentation. So this is the lesson 2, which is the fed batch culture. Okay, so as a sort of a review class, I will now I have posted in the VSUEE the questions that you have to answer, and it's not recorded, it's not graded by the way, but it's just a sort of a review in which you have to refresh your mind about the batch culture fermentation. Okay, so kindly answer those questions, or rather that one question, so that you can really refresh your mind about the batch culture fermentation process. So for the outcomes of this lesson for today, this you will be able to understand the concepts and principles of fed batch culture, and then to familiarize the uses, advantages, and disadvantages of the fed batch culture fermentation. Okay, so let us now start with the fed batch culture. Okay, so the characteristics of fed batch culture, it is similar to the batch fermentation process. Okay, so you will inoculate the microorganisms in a particular medium, and then the particular volume also, and then the volume of medium is inoculated with the organism and allowed to grow in a period of time. So the this will complete the fermentation process. But however, in the batch fermentation process, there are four microbial growth phases, so which is the lag phase, the exponential phase, the which is the constant phase, or the stationary phase, and then the fourth one is the cell death phase. Okay. However, in this fed batch culture, there is an extension of the batch culture by feeding periodically with medium with no removal from the vessel. Okay. So that is why it is called or named fed batch, because at a particular period of time after a complete set of the fermentation process, there is an additional feeding of the nutrients in order to extend the production of the microorganisms of a particular compound. Okay? So subsequently, a fed is initiated into the fermenter when a quasi-steady state is attained, in which the growth-limiting substrate has exhausted. Okay, with the microorganisms consumed. Okay, consume the nutrients in order to grow exponentially and also produce the particular compound. In the bioprocess or in the fermentation. So when the growth limiting with the substrate has already exhausted, okay, that's the time that there will be additional feeding of the nutrients in order to continue the microorganisms production or the microorganisms growth. That is why it's called the quasi-steady state, because there is a equilibrium of the growth rate of the microorganisms to the concentration of the volume of the substrate. Okay, the supply of growth limiting substrate or medium will sustain the microorganism's growth in the fed batch reactor. So it depends upon the substrate, okay, that will sustain the microorganism's growth in that reactor. Okay, so just like in the batch fermentation process, it starts in a low volume concentration. Okay, the initial concentration is low. Then the reactor will then continually be fed with medium concentrated with carbon and nutrients and nitrogen rather in increments. Okay, so after a particular period of time with the when the substrate has already been exhausted by the microorganisms, there will be a that's the indicator or the start that there will be feeding again of the nutrients, a particular volume, it, um, it depends upon the type of the fed batch process, whether the volume is fed constantly or there is an increment, there is gradual increase of the volume of the substrate to be fed at a particular period of time. Okay, so mostly the nutrients or the substrates are concentrated with carbon and nitrogen. Okay, so that's the basic needs or the basic requirements of the microorganisms for growth and metabolism. So, however, this type of culture it is still limited and it has still disadvantages because of the accumulation of toxic compounds as it dictates as it dictated the age of the microorganisms in the tank. Okay, so uh, as the microorganisms will continually grow and also um, consume the nutrients and then produce the particular compound that are in the medium. So it is still limited because the microorganisms age and it will really produce toxic compounds and secondary metabolites, So as what we have discussed from the previous lesson that there are two types of metabolites that the microorganism produce, the primary metabolites and then the secondary metabolites. So it dictates the age of the microorganism. So the production or the fermentation or the fed batch the fed batch process will still end because of this type of limitation. The toxic compound limits the continuous production of the microorganism. Okay. So this is the graph. Sorry, this is the graph of the or the illustration of the fed batch culture. Okay, so in the fed batch culture, you have this feedstock vessel in which it contains the nutrients or the substrates that are needed by the microorganisms, and then it will so it is still fed, but there is no removal of the compounds or there is no recovery yet here. Okay, so kaya after sometimes that the the fermentation or the fed batch fermentation has stopped, so there will be now a collection vessel that will then recover. There's then the recovery separator of the substrates and also the liquid medium, okay, at a particular period of time. But when the fermentation is still ongoing, so there is no recovery or separation in the fed batch process, but there is a continuous feeding of the nutrients. Okay, so let us now compare the batch process and the fed batch process. Okay, based on the biomass, based on the glucose, based on the product. As you can see here, class, that in the batch process, so there is a high concentration of glucose here at the onset of the fermentation. Okay, and as the microorganisms grow, it consumes the substrate. So there is now a decreasing supply or concentration of the glucose as the fermentation is going on. Okay, so that's the trend of the substrate, which is represented by the glucose itself. Okay, so there is a decreasing concentration of the substrate at a particular period of time until the substrate is already exhausted. But when the glucose has already is consumed, the biomass or the cells of the microorganisms are constantly increasing because no, so because it is exponentially growing, actively dividing as it consumes the substrate, and also the product is um, um, it is proportional to the biomass. The product is also increasing. Okay, so that's the characteristics based on the graph, based on glucose, biomass of product on the batch fermentation process. While on the fed batch process, as you can see here, since there is a constant feeding of the glucose, okay, so as you can see that in a particular period of time, there is additional feeding of the substrate, okay, so that's how it goes. There is the glucose, there is a particular period of time that it is fed and then consumed by the microorganisms. It will decrease and another increase again. So that's the graph or the trend of the glucose. And then also, well, the biomass, okay, it is constantly increasing. That's the yellow line. Okay, there is a constant increase of the cell concentration. And also, it is proportional also with the biomass that the product is also increasing. So that's the advantage of the fed batch process. If you want to produce large amounts or um, large amounts of product of a biological product, so we can use the fed batch process rather than the batch fermentation. Okay, so that's the difference of the batch and the fed batch process. Okay, so the advantages of the fed batch culture is that the maintenance of the fed batch culture is in a low substrate level. Okay, so the most problem of fermentation is when substrates are fed at a high concentration, repression occurs due to the high concentration of the substrate. There is a uh, uh, feedback inhibition when there is an increased concentration at the onset of fermentation. So that is why the microorganisms cannot produce um, the product, okay, because there is the microorganisms will tend to produce the particular kind of glucose of a sugar so that it will then produce the kind of product then. And then also the advantage also is the productive phase of the fermentation will be extended. As you can see in the graph class, that in the biomass, so it is constantly increasing. So there is a longer period of time that the microorganisms will not experience or will not achieve at a stationary phase. It is constantly increasing. Okay, so that's the advantage of the fed batch process. So the productive phase of the fermentation is extended. So there is a high concentration of the product that are produced. Okay, so there are many types of fed batch process. Later, we will discuss it. And in the cycle fed batch system, a periodic shift of the growth rate enhances secondary metabolite production. Sorry, as many secondary metabolites are not growth associated. Diba? So the secondary metabolites are produced. Okay, in the later stage of the fermentation. So the secondary metabolites are not that useful for the growth of the microorganisms. Okay, so the example of this is the kojic acid reduction. It is a secondary metabolite produced by the Aspergillus oryzae. So that's the perfect example of the kojic acid that uses or that employs or uh, that fed batch fermentation is employed in that kind of kojic acid production. Okay, and also many other secondary metabolites such as antibiotics. Okay, so another example of the uh, biological product that are produced through fed batch process. Okay, because the productive phase is extended. So another advantage of the fed batch culture is that the product formation in this system depends on the specific growth rate of the microorganisms. So the specific growth rate of the microorganisms are always achieved due to the constant feeding of the microorganisms due to and it also depends upon the concentration of the substrates that are being fed. Nutrients are fed exponentially. So there are kinds of fed batch process that nutrients are fed exponentially, meaning the nutrients are fed increasingly in increment. Okay, so there is at a particular period of time, for example, the concentration of the substrate is just like this, but in another period of time, so it is two times um, larger or two times of the volume that was fed on the first fed. Okay, so the nutrients are fed exponentially to obtain optimum desired products by monitoring the specific growth rate of the microorganisms. Okay, so this will be done always on a bioreactor. So if we the indicator that the substrate is already low is that there is a viscosity probe in the bioreactor so that it will then indicate the concentration of the cells that are being produced and also the biomass itself and also the volume of the liquid medium. So we can really, there is an indicator to that in the bioreactor process. So that's why it is a perfect timing. Of course, you have to first study your microorganisms and how you have to optimize the product, optimize the performance of your microorganisms on how it is being fed exponentially. Okay, so every 30 minutes every 1 hour. So we have to do a series of investigation on these microorganisms. Now, the example of this fed batch culture, as I have said, Karina, that it is the example of this is the antibiotic production, okay, which is mostly secondary metabolites. Okay, so in this fed batch culture, there is a drastic reduction of oxygen transfer and heat because of the high viscosity culture brought in the batch process. As you can see that it's just like it's still similar with the batch process, but the fermentation time is extended and the nutrients are being fed. So there is still a drastic reduction of oxygen transfer, okay, and heat. Okay, so because of the uh, because of the concentration of the high concentration of the substrate in the liquid medium. Okay, so using fed batch culture, the fed of the nutrients is controlled. Okay, so followed by the output of a certain volume from the fermenter. So it depends upon there is still a calculation to that and how to determine the concentration of the substrates in order to uh, be added in the bioreactor. Okay, so the technique reduces the high viscosity effect of the culture. So the example of this is that the biopolymer production usually encounters this problem, and feed batch culture is suited in the fermentation. So when there is so that there'll be in this process class, so the high viscosity effect of the culture is reduced because of the constant, because of the monitoring of the feeding of the nutrients. Okay, so it is highly controlled. Okay, for the extracellular enzyme production, so it is controlled by the catabolite repression, meaning so when there is an enzyme, there is too much enzyme in the medium, so the enzyme itself will then repress the production of the microorganism. So enzyme synthesis is inhibited by the presence of readily utilized carbon source on inhibitory carbon source. So using the fed batch culture technique, the inhibitory carbon source is fed gradually. Okay, so to overcome the inhibitory effect for the production of extracellular enzymes. This overcomes the limitation of the catabolite repression, the which is the inhibition of these microorganisms in an extracellular enzyme production. Okay, the presence of the utilized carbon source or the inhibitory carbon source are accumulated when there is already enough enzymes. So when there is when the fed batch culture process is employed, so the carbon source is fed gradually. Okay, so there is an exponential feeding of the carbon source in order to overcome the inhibitory effect of the uh, in the process. Okay, for a constant production of the extracellular enzymes. So as what I have said kanina, that there are two classifications of fed batch culture. One is the fixed volume fed batch culture, and then the second one is the variable volume fed batch culture. Okay, in this fixed volume fed batch culture, so the limiting substrate is fed without deleting the culture, meaning the substrate is very concentrated. Okay, it is in an undiluted form, and as um, as the name implies that it is fixed, so the feed is provided at a constant rate. So every 30 minutes, so there is a constant rate in the feeding uh, of the substrate in the bioreactor. Okay, the production of the mass of biomass per mass of substrate is constant during the fermentation time. And then this consider a batch culture in which the growth of the process organism has depleted the limiting substrate to a limiting level. So yeah, that's when they since the microorganisms microorganisms is uh, fed in a very concentrated liquid and also in a constant rate, so there is also a constant increase of the production of the biomass and also the constant production of the product. Okay, so this consider a batch culture in which the growth of the process organism has depleted the limiting substrate to a limiting level. So when there is now a depletion because of the microorganisms consumption of the substrate, so the substrate is like the substrate itself is the limiting factor to the production of the product by the microorganism, and that's also the time that it will then another concentration or another the, the substrate is being also mixed or added in the fermentation vessel. Okay, so there is an indicator to that in the bioreactor. So how about with a variable volume fed batch culture? So in the fixed volume fed batch culture, so the volume of the substrate is fed at a constant manner and a constant concentration. Okay, while here the volume changes with the fermentation time due to the substrate fed. Okay, the substrate is provided in a manner that maximizes the specific growth rate. In this fixed volume fed batch culture, so the specific growth rate or the maximum growth rate is not that achieved. But here in the variable volume fed batch culture, so the goal of this classification of fed batch is that it maximizes the specific growth rate. So there will be increments of the feeding of the volume of the concentration of the substrate. The substrate is added exponentially for biomass production to maintain constant growth rate in a culture growing exponentially. Okay, the substrate is exponentially added in order to have a constant growth rate, the maximum specific growth rate in a culture growing exponentially. But there is a limitation to this area, which the conditions of the variable fed batch culture are the input is equal to the consumption of the cell. The input of the substrate should be equal to the consumption of the cell. And then the input of the substrate depends on the specific growth rate and the total concentration in the culture. Okay, there is a calculation to that. Okay, I guess that's the last slide. Okay, so again, there are critical conditions on the variable fed batch culture in which should know the input of the substrate should be equal to the consumption of the cell in order to achieve a maximum specific growth rate, and also to really to achieve a constant growth rate in order to have a constant maximum production of the product. And the input of the substrate depends also on the specific growth rate and the total cell concentration on the culture. Okay, that's the fed batch culture fermentation. Okay, as for the learning task class, please answer these questions. That's it's all for the discussion. And if you have any questions, please don't hesitate to chat in the group chat, in the Facebook Messenger, or also you can email me, and also you can directly chat or message in the VSUE. Okay, for the learning task class, this will be your the things that you have to answer. So because when we meet again, you will answer this orally in the class, okay, after the midterm examination, so that we can have a review of our discussion. So for the learning task, you have to explain the principle of fixed volume and the variable volume fed batch culture using the graph shown below in your own words. Okay, so you cite the references used for the explanation. This is the explanation or the characteristics based on the characteristics of the specific growth rate, the biomass concentration, and the substrate and the growth limiting substrate over time on the fixed volume fed batch fermentation. And this is also the graph for the variable volume fed batch fermentation. Here to explain why the this is the trend of the graph and how it relates to the biomass concentration, specific growth rate, and other substance. The same with this second graph. Okay, this topic is included in the midterm examination class. So please watch this video. So this is again another video that you can watch. It is in YouTube, so please don't miss this watching this video for another supplementary discussion. Here is it again. Okay. Thank you so much class for watching, and I hope you have learned much about the fed batch culture fermentation. Thank you.